Yeah, thanks for the introduction. And I would like to thank Alan again for inviting me to this meeting. So I'm Junchen, and I'm currently doing my PhD in the Lund University about some symbiotic bacteria in Hemipteran insects. But today I'm gonna introduce my master's research on the common endosymbiotic bacteria that lead to reduced genetic diversity in the common blue-tail or blue-tailed damselfly, the Ischnura elegans. So I will tell you what is endosymbiosis and what is genetic diversity and what we have done in the blue-tailed damselfly. So genetic diversity, we talk a lot about genetic diversity today. So it describes the range of variation in genetic information present in the population of a species. So why genetic diversity matters? Because genetic diversity is the raw material for nature selection. So Charles Darwin developed this theory, nature selection theory, in 1850s. But at that time, people knows almost nothing about DNA. So the genetic heredity only developed after 1900. But now we know that those variations that we see are actually genetic variations. And the nature selection actually describe the change of frequency of genotypes in a population under different environment. So we can imagine that there are two genotypes in a population, so genotype 1 and genotype 2. If genotype 1 is fitter than the genotype 2, then the genotype 1, then the selection will favor genotype 1. So the individuals that carry genotype 1 will produce more offspring. So in the next generation, there will be more individuals of genotype 1. So the frequency of genotype 1 increase. So this is how nature selection works. And the genetic diversity is linked with the survival of the species. You can imagine that if there is no genetic diversity, then the species, the nature selection won't happen, and the species cannot to adapt to the changing environment. So there is a very classic examples of how low genetic diversity linked to the survival of the species. So we all eat bananas, and we know that. So the banana type currently in the supermarket is called Cavendish, but actually 100 years ago, the dominant type is called Gros Michel. So how Gros Michel just disappear suddenly? So actually, it is because a disease called the Panama disease. Because how people grow banana is that they have a plantation, and almost all bananas individuals are the same. So they are actually genetically similar to each other. So when the disease comes, the banana plantation and also many other crop plantation is like a wonderland for those diseases. So if the disease infects one individual, so basically it will infect all the individuals. So that's why the whole population will collapse. And this is how Gros Michel just disappeared suddenly. And luckily, the Cavendish has a gene that can help them to be resistant to this disease. But recently there is a new strain called tropical race 4 that is also that is threatening the Cavendish banana, and maybe in the future we won't see Cavendish in the supermarket anymore. And genetic diversity is also talk a lot in the conservation biology. So those endangered species usually they have a very small population. And a problem with those small populations is that the individuals within those populations that tend to be related to each other. So this will cause a problem of inbreeding. And inbreeding will cause inbreeding depression, and which will cause the reduce fertility and also reduce survivability. So the population of those endangered species is very vulnerable. So genetic diversity is very important, and we should keep them at a certain level. And there are many factors that can affect genetic diversity. So the monoculture we have talked about, and also human activities such as deforestation, which will cause habitat fragmentation and habitat loss, and also species migration and population range expansion. So when a new species, when a species try to colonize some new habitat, only part of the population will migrate. So this will potentially cause a population bottleneck. And also climate change, which will also cause habitat loss and also directly impact on the population. And those factors are what we can observe easily. But there are also some hidden players that can also cause reduced genetic diversity, and they are not easy to be spotted. So those are the microbes like virus, bacteria, or fungus that causes the epidemics. And those epidemic diseases will also reduce the population size and also cause selective spread, which I will explain later. And this has something to do with the bacteria that I will introduce today, the Wolbachia. So before we go into Wolbachia, I will first introduce the symbiosis. So symbiosis describe a close and long-term interaction between two organisms. It can be mutualistic, so both sides will benefit from each other, or it can also be parasitic. Sorry, and there is a more intimate type of symbiosis called endosymbiosis that happen only within the cells of organism. And for a classic example, you can think about mitochondria, so the powerhouse in our cells that produce energy. So mitochondria was a free-living bacteria, and it was later, like several billion years ago, it was taken by some other free-living cells, and then they together evolve into the eukaryotic cells that we bury today. So Wolbachia is an endosymbiotic, so it lives inside the cells of the host. It is a facultative symbiote, which means that it is not essential for their host to survive, and it can sometimes jump between hosts. Wolbachia is maternally inherited, which means that they can only be passed from female host to their offspring. And the Wolbachia is one of the most common endosymbionts in insects. There are many studies estimate that Wolbachia infect around 20% to 70% of insect species. So we just see that Wolbachia is very common. They infect a lot of insect species. But apart from insects, they can also infect some other arthropods and also nematodes. So Wolbachia can do a lot of things to their host. For example, they can affect the fecundity, the survival of their host. They can make their host more or less resistant to pathogens. And some Wolbachia strain actually steal some genes from other bacteria, and they can produce some nutrients to their host. And some Wolbachia strains can change even the behavior of some of the host species. So they can do a lot to their host. But what is the most intriguing things about Wolbachia is that they can actually manipulate the host reproduction. So, for example, they can do feminization basically change the gender of the males, and they can cause parthenogenesis, which means that the unfertilized eggs can survive and produce offspring. And for some male Wolbachia strains, they will kill the males in a population, so make the sex ratio in a population highly biased towards females. And there is another very common thing about Wolbachia is the cytoplasmic incompatibility. So basically, the uninfected females cannot produce any offspring with the infected males. So I will give you a very like classic examples that maybe you have heard a lot recently. So recently on the news, there are many news about like releasing mosquitoes around the world, and it's getting more and more common recently. I would say it happens like explosively in recent years, and this has something to do with Wolbachia. So the mosquito species, the Aedes aegypti, it is the main carrier of many lethal pathogens. So how people combat those pathogens using Wolbachia is that there are two ways. So first, they just inject the Wolbachia, to just infect those mosquitoes with Wolbachia, and because Wolbachia can compete with those pathogens. So for some strains, they are very powerful. So the Wolbachia will just outcompete those viruses like dengue and Zika. And for another method, the scientists just infect the male mosquitoes, and as we have seen that the uninfected females cannot produce viable eggs with the infected males. So basically, those eggs will die because of the cytoplasmic incompatibility. So in this way, the Wolbachia will reduce the mosquito population size and stop the spreading of those lethal diseases, and which turn out to be very efficient. So for our research, we interested in how Wolbachia reduced genetic diversity of insects. So they did this by the selective sweep. So what is selective sweep? So we can imagine a host population with some genetic variations maintained in this population. And if some of the individuals are infected by Wolbachia, and if the Wolbachia can deliver some benefits to their host, for example, they can make their host, they can allow their host to have more offspring, or they can improve the survivability of their host. And then the infected host, after selection, the infected host will probably have more offsprings. And then those infected individuals will be dominant in the next generations. And this process can happen very quick. So that's why we call it a sweep. So we studied the Ischnura, and we have seen that the Wolbachia is very common in insects. But before my study, there are not many studies about how frequent Wolbachia infects the Ischnura species. So for my research, I want to know if the Ischnura species if the Ischnura species really infected with Wolbachia, and if so, how frequent does the infection happen, and also how diverse are the infections? And then the second question is that, can we find any evidence of Wolbachia-induced selective sweep in Ischnura populations? And the species that we focus on is Ischnura elegans, which is a very common species. And some interesting about Ischnura elegans is that it's the female of Ischnura elegans is polymorphic in colors, so it has three color morphs. And there are some quite interesting evolutionary questions related to these color morphs. But I won't talk too much about it. And if you are interested, you can look for some researchers done by my previous supervisor, Eric Swanson. So he has studied this species for more than 20 years. So he knows a lot about it. So for us, so this species is very common, and it has a very wide distribution. So this is the map that I take from the GBIF website, and basically you can observe this species across Europe and Asia. So it's fairly common. So what we did is that we focus on the Ischnura elegans, and we collect samples mainly from Sweden. And we also have colleagues from other countries that gather specimens for us. So we have a kind of decent collection of samples from across Europe. And apart from Ischnura elegans, we also study several other Ischnura species like Ischnura pumilio, which is a sympatric species to Ischnura elegans. So basically, it means that they share the habitat. And also we have some specimen from Ischnura genii, so which is called, and they are called island blue-tailed, I think. So you can only find them in those islands. And then we also have one specimen from Morocco, so the Ischnura saharensis. So to screen Wolbachia, we sequence some genetic markers from Wolbachia. And for the host genetic diversity, we also sequence several medical genes. And also later we have sequenced also one or two nuclear markers. So this is what we found. So here is the map showing the infection levels in different populations. So the side of the pie chart indicate the side of the sample size. And different color indicates different Wolbachia strains. So here you can see that we have found three Wolbachia strains, W81, 2, 3, in Ischnura elegans and also in Ischnura genii, the island blue-tail. And also we found two other divergent Wolbachia strains in Ischnura pumilio. And all Ischnura pumilio samples from Sweden are infected. And actually, Swedish population is highly infected by Wolbachia. And the presence of the W81 strain in Ischnura elegans and Ischnura genii indicate that the Wolbachia was potentially horizontally transmitted between those two species. And here from this map, we can see that there is a trend that the North Eastern European populations are heavily infected with Wolbachia, especially with the strain W81. And in our study, we also estimate the date of the infection. So it was around 10,000 to 40,000 years ago. And this indicates a potential infection wave that happened after that happened along with the post-glacial range expansion of the Ischnura elegans. But unfortunately, we don't have any samples from the Central Europe. So in the future, it would be nice if we can increase the sample size from those populations and also get some samples from the rest of the countries, so we can have a broader pattern of Wolbachia infection in Ischnura elegans and some other Ischnura species. So what about the genetic diversity? So how Wolbachia infect the genetic diversity? So this is a haplotype network. So haplotype is basically a combination of a set of genotypes. So in this graph, it is colored by the so each circle represents one haplotype, and the black bar between two circles indicate the distance between two haplotypes. So if there are more black bars, then they are more divergent, those two haplotypes. And this graph is colored by the countries. And first, we can see that the haplotype 1 is very common commonly seen in many European populations. And if we look at the Cyprus populations, it turns out that it maintains a very high genetic diversity because it has a lot more haplotypes. And if we look at the infection, so this is so they are the same haplotype, but the second one was colored by the infection. So we can see that the different strains, different Wolbachia strains, was linked with different haplotypes, and also the WLE1 strain is mostly linked to haplotype A, and the WLE1 also linked to a very low genetic diversity because apart from haplotype A, there are only like two or three haplotypes linked with this infection. But if we look at all those uninfected populations, we see that there is a much higher genetic diversity in those uninfected ones. So actually, this is quite contrasting. So the significantly reduced genetic diversity in the WLE1 infected population actually indicates a Wolbachia-induced selective SWIP in the Ischnura elegans populations. So this is basically the main findings we have from our research. So what does this result mean to Ischnura elegans? So we know that Ischnura elegans is undergoing a northern range expansion in response to climate change. So probably the reduced genetic diversity will limit the adaptation of Ischnura elegans to new habitats. And recently, I also checked the report from 2021 and from in Britain. So it indicates that the population of Ischnura elegans is actually declining because of the loss of habitats and also pollutions. So the low genetic diversity will probably make the survival of the population harder. But on the other hand, could the Wolbachia also be a novel weapon for Ischnura elegans to cope with climate change? We don't know. So more studies are needed to study the functions of Wolbachia infection in Ischnura elegans. So for this study, I want to thank my previous supervisors and employee. So she's now a teacher in Helsinki University. And then Eric Swanson, so he's now still a professor in Lund University, and also some of my colleagues. And if you want to know more about this research, you can check this paper we have published on BMC Ecology and Evolution. And if you want to know more about my current research on symbiotic bacteria in hemiptera insects, you can check this website. Yeah, so and also if you want to know more about the evolution of the color polymorphism, you can contact Eric, and I'm pretty sure that he will be happy to share it with you. Thank you very much.